Okay, so there are one or two other details I wanted to share with you regarding the structure of DNA. And the first is to do with its stability. DNA is an incredibly stable molecule. That means that it can remain intact for a very long period of time. Okay, it's not prone to degradation, to breaking down. Okay, I'm going to share with you a little clip from a podcast from Inside Science where you can hear just how stable it is. What we found is that actually the last part of you to rot away isn't your bones at all, it's your DNA. And if you're buried or your body ends up somewhere where no one can find it, and it gradually breaks down, then what happens is that the DNA is the last thing to break down. It's incredibly stable, much more stable than bone. And this comes mostly from the hydrogen bond. So DNA is a very long molecule, and there are many, many hydrogen bonds within it. They're sort of colored gray here in this diagram, okay? So if we look at just a short length of DNA, like we've got here with the PNS representing the sugar phosphate backbone here, and you just look at one of the short sections, you can see just this incredible number of hydrogen bonds because there are 2 between every A and T and 3 between every C and G. So that adds up to a lot of hydrogen bonds. And as you remember from water, hydrogen bonds, although they are weak bonds on their own, when there are lots of them, the combined strength is very high. Okay, so they are weak bonds. Don't make the mistake of saying strong hydrogen bonds ever, but there is strength in numbers when it comes to hydrogen bonds. So slightly beyond A level, there is another reason why DNA is such a stable molecule, and that's to do with something called base stacking. Okay, there are van der Waals attractions between the stacked bases. So you can see here in this diagram, just to remind you that the bases are stacked on a tilt. This is how they're stacked because the molecule twists as it goes up. And what you get is an interaction between bases stacked one upon the other. So the base stacking is nicely illustrated with this model here. The gray represents the sugar phosphate backbone, and the blue and kind of coral colored balls here represent the complementary base pairs, and they're paired off. Okay, so if we look at it sort of from the top, we can see this larger base here, so that would be a purine, and then that's paired with this smaller pyrimidine here. Okay, but then each complementary pair is colored in kind of blue or coral, and you can see they're not stacked directly one above another. There's a slight rotation as you go down, but more importantly, they're very closely associated, one base pair to the next. Okay, and there are these forces of attraction between the base pairs. There's base stacking, as we call it. Okay, and this contributes to the stability of the molecule because not only is there an attraction there that helps to hold the structure of the DNA molecule together, but they because of the close interaction, there's no space for water here. This is a completely hydrophobic region. Okay, so within the double strand, there is no water. And that's important because it means that there is no dissolved ions either. So no danger of free radicals, oxygenizing or reducing agents getting in there and reacting with the bases. The bases are protected inside this double helix. And you can write that the double helix offers protection, and bases are on the inside of the double helix and they're protected. Okay, part of that protection comes from the fact that this whole region is hydrophobic due to the way the bases are stacked one on top of the other here. Okay, of course, they're still susceptible to stuff that doesn't need to go through water. So gamma radiation, UV radiation, as you know, can damage the DNA bases. But as far as possible, the bases are being protected by this structure, by being inside like this and being stacked together with interactions between them in a region that's completely hydrophobic. So base stacking and a large number of hydrogen bonds give DNA this incredible stability that the molecule has. The other thing that you need to be aware of is that the DNA molecule comes in two forms, okay? You've got the linear DNA that you find in your own cells, in eukaryotic cells. Linear in this sense, it doesn't mean in a line, it just means it's got two ends, okay? There's a terminal and then the molecule goes through in a line until it reaches the other terminal, okay, up here, you see. Whereas in prokaryotic DNA, the DNA is circular, there is no end to it. All right, so this is a DNA from a prokaryote here, and just to show that it's circular. All right, in all other respects, it's identical. Okay, this is the same 4 bases, and when we get to looking at genetic code, you'll find that the code is the same. Okay, but you have this circular DNA that you find in prokaryotes and this linear DNA that you find in eukaryotes. Now, there are other differences, but they're to do with function. We're only interested in structure here. When you look inside in a prokaryotic organism, you will tend to find one chromosome, okay, one large chromosome, and then lots of smaller rings of DNA, which we call plasmids. Okay, now this is a difference to the way that the eukaryotic DNA is found. Eukaryotic DNA is found in multiple chromosomes, and that's housed in a nucleus. There is no nucleus in a prokaryote, and instead the DNA is free floating. It's also naked, right, in the sense that it's not wrapped around histones. Eukaryotic DNA is wrapped around histones. Prokaryotic DNA is not. Okay. When we talk about prokaryotic DNA, we tend to say there's one large chromosome and then smaller plasmids, but the chromosome is only large in comparison to the plasmids. It's very, very short in comparison to eukaryotic DNA. Eukaryotic DNA is much larger, okay? Eukaryotes tend to have a lot more genes than prokaryotes, all right? I want to say too much about that because we don't want to talk about function at the moment, and I don't want you thinking, oh, that means that prokaryotic DNA is in some way less advanced or less involved or inferior to eukaryotic DNA. It's not that at all, okay? But we don't want to go into that at the moment, all right? One of the big differences between the prokaryotes and the eukaryotes is that the only way to for a cell to get DNA in eukaryotes is to inherit it from its parents. Okay, that is not true for the prokaryotes. Okay, with the prokaryotes, they are able to trade in plasmids. Okay, so if you're thinking about the genome so the genome is all the DNA in the organism. If you're thinking about the genome of prokaryotes, you tend to have to think about the entire population, not the individual, because these plasmids are traded. Okay, so an individual doesn't have much DNA, but the population might have a considerable amount. So this is a difference that you need to know about. Prokaryotic DNA is circular and eukaryotic DNA is linear. Notice the prokaryotic DNA is double-stranded. Now you might think, why am I making the point about that? The amount of kids in year 13 who start thinking that prokaryotic DNA is single-stranded for some reason. It is not. It is the same stuff. It's double-stranded, sugar phosphate backbone, adenines, thymines, cytosines, and guanines. It's just that rather than there being terminal ends, there are no ends. It's circular. Okay.